Happy day everyone. In the last video we have completed about the epidermal tissue system. In that we have talked about the epidermal cells. Now we will further start with the stomata. The stomata are generally found in epidermis of leaves. They have something called as guard cells which are tube bean shaped and they encloses the stomatal pore. That is, within the guard cell there will be stomatal pore. And then talking about the grass. In case of grass, the shape is changed to dumbbell shape. They are generally tube bean, but in case of grass it is dumbbell. Okay, and talking about the layers, the layer which is closely associated to the stomatal pore is called as the inner layer, and they are thick, which is away from it will be called as the outer layer, and which is thin. Okay, these guard cells consist of chloroplast and they also perform the opening and closing of stomata. And the subsidiary cells are nothing but some cell due to the effect of the stomatas. They will some cells or epidermal cells, due to the effect of it, they will be getting specialized. These specialized epidermal cells are called as subsidiary cells, and the stomatal apparatus consists of stomatal aperture, guard cell and subsidiary cell. So now let's talk about the hair. To talk about the root hairs, they are generally unicellular and they absorb water and minerals. But in case of stem, they are called as trichomes and they are also multicellular. These may be branched or unbranched, they may be soft or stiff, and they also help in preventing the water loss due to transpiration. So with this we are completing the epidermal tissue system. We talked about epidermal cells, stomata and root hair. So to talk about the ground tissue system, except epidermis and vascular tissue, all the other tissues are ground tissue system. They consist of simple tissues, and in the ground tissue system we have parenchyma which are present in the cortex, pericycle, pith and medullary rays. In case of leaf, we have something called as mesophyll cells which have thin walled chloroplast. So now let's move to the vascular system. Okay. In this vascular system is consist of xylem and phloem. This is together called as the vascular bundle. So vascular bundle refers to them. And we have two types, which is in the dicot system we have something called as cambium. Okay, this cambium will be promoting the secondary growth in the xylem and phloem, and so it is called as the open vascular bundles because they are open to secondary growth. But in case of monocot, there is no cambium because there is no secondary growth in the monocot, so they are called as the closed vascular bundles. So in case of roots, the xylem and phloem, that is vascular bundles, are arranged in different radii. Okay, but in case of stems and leaf, they are arranged in the same radius, which is called as the conjoint. In case of root, it is called as radial. And in case of this conjoint, this phloem will be always the outer side of xylem. That is, if this is the cell, if you see here, this will be xylem, the outer side will be phloem. So this is how they will be arranged in the conjoint cell. So still now we have talked about the tissue system, epidermal, ground tissue system and vascular. Now we will be talking about the dicotyledonous root. So to start with the epidermis. The epidermis is the outermost layer of the dicotyledonous root, and as we already discussed, they will be having the root hair, right? So the root hair, and they are unicellular root hairs. Okay. Next to that we have the layer called cortex, which is filled with parenchyma and also with intercellular spaces. In case of simple tissue, we talked that parenchyma may or might not contain intercellular spaces. So here the parenchyma with intercellular space. So next to that we have the layer called endodermis, which is the inner layer. These are the single layer without any intercellular space, and they have an outer covering which is made up of waxy material subberine, which is also known as the Casparian strips. Okay, you can think it as the outer covering of the endodermis, and this is called as the Casparian strips. And talking about the next layer, after to endodermis, we have something called as pericycle, which is a thick wall, and they also consist of the parenchymatous cells. And in the pericycle, it will be promoting the secondary growth, that is lateral roots and vascular cambium. These are the major things for the secondary growth. And in case of the dicotyledonous root, the pith is very small. Now, to talk about the conjunctive tissue, which is nothing but these are the tissue which present between the vascular bundle, the xylem and phloem. So in case of dicotyledonous root, it generally there are only 2 to 4 xylem, and the phloem will be arranged in the patches. Okay, after the lateral stage, when they are getting into the secondary growth, there will be a formation of the cambium ring, so that I have mentioned as later. So Stele is nothing but the pericycle, vascular bundle and pith. That is, all the layers which are enclosed within the endodermis are called as stele. So this is about dicotyledonous root. Monocotyledonous root, they also have the same layers as we discussed in the dicotyledonous one, but we have some few differences. Let's see that. Here the xylem will be more. Okay, if you see, in case of dicotyledon root, we said only 2 to 4 xylem will be present. But here, usually, there will be more than 6 layers. 6 xylem will be present, which is called as the polyarch. Okay. Then talking about the pith, here we talked it will be very small. But in case of monocotyledonous root, it will be large and also well developed. Okay. And in monocot, there is no secondary growth. In dicot root, there will be a secondary growth. Okay. In the next video, we will be continuing with the dicot stem and monocot stem. Thank you.